Now, the biochemical and cellular background of the viruses. What is the biochemistry of the viruses? What is the biochemistry of the virus structure and the ingredients which are present inside the virus? Biochemical and cellularly, actually, this is the different viruses looks like. This is just a diagram in which we can see some of the viruses which are including the envelope of the viruses and how they look like. On the left side of the viruses, they have the spikes. Some of the viruses, actually, these are the envelopes of the viruses. This is the Wendell M. Stanley, who actually for the first time got the Nobel Prize in 1946 for discovering the biochemistry, which was involved in the tobacco mosaic virus. Tobacco mosaic virus is the helical type of the virus, just you can see in the diagram. So he systematically investigated its biochemistry, that how biochemically the virus is working and virus is acting on the host. Symmetry of the different viral capsids. Actually, as we already studied, that virus can be the helical shape, which we take the example of the Ebola virus, and then it can be the icosahedral. Icosahedral has different the corners. Here we give the example of icosahedral can also be the rounded shape. Then they may be, it is with different corners, maybe the 20 corners, maybe 32 corners. Similarly, there are some complex kind of the viruses which included both of them. They have the helical shape and also have the, just like the bacteriophages. The capsid of the bacteriophages is surrounded by the helical symmetry, and then the surface of the bacteriophages have the helical symmetry, and the capsid has the icosahedral symmetry. There are some enveloped viruses and some are non-enveloped viruses. The capsid of the virus, which is surrounded by some envelope, those are called enveloped viruses. Most of them are naked. This is the capsomere, and then the spikes already present in the influenza virus. You can see the diagram shown by the electron microscope. An enveloped helical virus, which is shown in this diagram. Now we can take just a little survey of different human viruses. Non-enveloped viruses may be the single-stranded viruses or the double-stranded viruses. Parvovirus is the single-stranded and non-enveloped virus, which is causing the diarrhea in the human beings. Then the papovavirus, it is double-stranded and it is also non-enveloped. Then adenoviruses are also double-stranded viruses and very important from the human point of view. And then the iridovirus is also double-stranded virus. So from the non-enveloped viruses, only the parvoviruses are from humans' point of view are important, and they are single-stranded non-enveloped viruses, while all other three species are the double-stranded viruses. Then the enveloped viruses, HEPA-DNA virus is the most important, that is partially double-stranded DNA. And then pox virus was that such kind of virus against which for the first time the vaccine was developed. And then herpes virus, again the double-stranded DNA. Then the non-enveloped viruses, single-stranded picornavirus. Picornaviruses are very small viruses. It is the example of the very smallest viruses existing on this earth. They are single-stranded RNA. Then reoviruses are about 100 nanometer in length, and they are also double-stranded RNA, and they are non-enveloped viruses. Then enveloped viruses all are single-stranded RNA viruses. They are rhabdovirus causing the rabies disease, very common disease, and they are single-stranded RNA. Then Toga virus, orthomyxovirus is also very important, and there is also single-stranded RNA. Then Bunia virus, coronavirus are the examples of some big genera of the species of the viruses. Paramyxovirus is also single-stranded RNA. Then retroviruses and arenaviruses are also single-stranded and enveloped viruses.